Hello everybody, I hope you've had a nice weekend and getting ready to jump right back in again. This week we're finishing off plant transport, so we're finishing off this section in the multicellular organisms unit. We've got 3 different learning intentions today. We're learning about the factors affecting transpiration, we're learning about measuring transpiration rate, and we're learning about the number of the stomata on the leaves. So let's take a look at the factors affecting transpiration. We know from last week that the stomata are tiny pores or holes on the bottom of the leaves. They live in the lower epidermis. Their function is for gas exchange. Okay, so during the process of photosynthesis, we need carbon dioxide to come into the leaf and oxygen diffuses out of the leaf through these pores in the lower epidermis. We also know that during transpiration, so the movement of water through the plant and evaporating through the leaves, we know that the stomata are involved in that evaporation of water vapour as well. So if we are to think about transpiration as us hanging out our wet washing outside, okay, so take a towel for example, we need to think about what conditions outside are going to be the best for that towel to dry the quickest. So we're going to wait until it's nice and sunny outside, so there's a warm temperature. There's maybe a nice breeze in the air, so it's a wee bit windy, and there's low humidity, so there's not a lot of moisture in the air. So humidity as in when you go on holiday and it feels quite sticky and yucky and too warm outside, like if you're in Florida, for example. You need a good thunderstorm to clear the air. So that's what we mean by humidity, so moisture in the air. The same way that we think about our towel drying the best, we also think about the factors that can affect the transpiration rate. To increase the transpiration rate, we want to increase the surface area of the leaf. So we want to make sure that that leaf's nice and big, because the bigger the leaf, the more stomata in theory there should be. We also want to increase the wind speed, so we want it to be windy. So the more wind there is, the more water vapour is picked up by that wind and carried away. And if we increase our temperature, the higher the temperature, the hotter it is, the more evaporation is going to occur. Looking at the other way, we're looking at factors that decrease the transpiration rate. So humidity. The more humid it is outside means there's more moisture in the air. If there's moisture in the air and moisture in the leaf, the water from the leaf has got nowhere to go. Okay, so there's going to be less transpiration happening when it's humid outside. So if we take an experiment and we're looking into decreasing the levels of transpiration, if we were to put plant A, if we were to have oil on top of the leaf in plant A and we were to have oil on the bottom of the leaf in plant B, which one would have a decreased level of transpiration and why? Think about where are the stomata located. I'm going to give you 5 seconds to come up with an answer. Okay, so having had a wee think about that, the answer would be plant B. Plant B will have decreased levels of transpiration because the stomata are located on the bottom of the leaf, and if we are to cover the bottom of the leaf in oil, then the pores are going to get clogged up with the oil and the water can't evaporate out. Okay, now that we've looked at transpiration, the factors affecting transpiration, we're going to look at measuring transpiration rate. So this image has been used in SQA documentation. It's on their website. In measuring transpiration, we're looking at, as the plant transpires, we're measuring the coloured water being drawn up through the glass tube, and we're going to measure the distance the coloured water has travelled within a set period of time. So for example, if we were to leave this experiment set up for 10 minutes, how far has the water travelled, or what volume has been drawn up. So taking light intensity as our measured variable or our independent variable. We know from our knowledge that light intensity, so the more light that there is, the stomata are open in the daylight, which means evaporation can occur and gas exchange can occur. In low light intensity, there will be less transpiration taking place. So linking that to the experiment, it means there'll be less volume taken up. As we increase the light intensity, what we expect to see, and what we do see based on this table, is that more volume is taken up. So the higher the light intensity, the higher the rate of transpiration. Looking at another factor that affects transpiration, wind speed. The faster the wind speed, the more volume is taken up, which means increasing wind speed will increase the rate of transpiration. And you could potentially set up another experiment in relation to temperature and look at how increasing temperature affects the rate of transpiration. And lastly, we're going to look at the number of stomata. The number of stomata on the leaf depends on the conditions that the plant has been grown in. If there are large numbers of stomata on the leaf, we expect the transpiration rate to be higher. Okay, but that depends on the conditions that the plant is grown in. Okay, now when we're talking about the number of stomata, you may also be asked in an exam question about calculating the number of stomata. So for example, if this particular plant species A, if this particular section was 1 millimetre of a plant, okay, and the question asks you how many stomata would be present on 2 centimetres of a leaf. So what you need to work out is, we know that 1 millimetre has 6 stomata based on the picture, but then we need to work out, well, in 1 centimetre there's 10 millimetres, so in 2 centimetres there's going to be 20 millimetres, which means we need to times 6 by 20, which means we'll have 120 stomata on 2 centimetres of that leaf. Okay. So like we said, the environmental conditions that the plant grows in will affect the number of stomata. So if you have a plant and you want to grow it in the desert, okay, so you're out in the Sahara Desert, looking at the level of water that's available there is really low. There's not a lot of water in the desert. So the number of stomata per leaf in plants in the desert should be low, because we want a low evaporation of water. We don't want to lose water. The plant needs water, so we want to keep it. So looking at the picture of the cacti, we want to be able to conserve as much water as possible and not lose it. Okay. On the other hand, if you've got moist conditions, so in the Amazon, for example, there's lots of water available. Okay, so in theory, there's lots of water available, which means we want to have lots of stomata so we can get rid of that water that we don't need. So you want a high transpiration rate. However, you may be presented with something like this and have to explain why in the desert there might be a higher transpiration rate, because there's dry conditions, there might be high winds, there will be high temperatures. So high temperatures, low humidity, and the high wind speed are all factors which should increase evaporation of water, but it's the adaptation of the plant itself, so the plant being able to grow there, that causes the evaporation to be low. Alright, and then in moist, humid conditions, so we know there's lots of water available, which means we want the water to get out because we're taking up too much. So if we look back to the factors affecting transpiration, if we increase the humidity, in the Amazon, it's a rainforest, it's a very humid place, the humidity is really high, which means the transpiration rate should be low in theory if we look at that. So there may be some other factors at play other than the environmental conditions that it plays in. There could be adaptations of the plant and things like that to consider when talking about evaporation of water. Adaptations of plants come up in Unit 3. So in an exam question, you may be asked to talk about how the adaptation of a plant prevents water loss, so it prevents this transpiration happening. So you're linking your knowledge throughout the course instead of just focusing solely on the little bit of Unit 2. They want to see, the SQ want to see you be able to paint a bigger picture and be able to analyse it from every angle of your knowledge within the SQA documentation. I hope that makes sense. So, to sum up today, we've looked at the factors affecting transpiration, we've looked at measuring transpiration rate, and we've looked at the number of stomata. So there's now a worksheet on the team's assignment page. Again, complete the answers on that. Most of it are past paper questions which have been taken out using the snipping tool. So to fill this in, you will need to create a text box and put your text box over the top of those answers. I've saved this. If you've got trouble loading it or downloading it from Teams, please let me know and I'll email you a copy. You can also use, again, page 25 of the multicellular organisms revision notes. This will help you with the task, and I've also included the BBC Bite Size page to help you consolidate your knowledge, and there's a nice wee video on there as well that kind of sums up the whole topic. Again, same as always, if you need anything, please let me know. I've also included on the assignment page of the hours that I'm going to be available every day, so it means that you've got set times to contact me rather than waiting for a few hours or days before I get back in touch with you. You've got a set time, you know I'm available for you. But again, if you need anything, just please let me know, and best of luck.